Hi Christof, you're presenting a poster here at HPLC conference. What is it about and why is this topic particularly relevant right now?
So this poster is about the comprehensive analysis of adeno-associated viral vectors, leveraging anion exchange chromatography and size exclusion chromatography. The topic is very relevant these days as we see an emerging need for gene therapies that often use adeno-associated viruses as viral vectors to deliver the genetic information to the target tissue. For this work, two different columns have been used, the Thermo Fisher Scientific ProPack 3R SAX column and the Thermo Fisher SurePack Bio 550 SCC MDI column. Both columns are leveraging a 3-micron particle design, which is monodisperse. So that means that each particle is more or less the same size, and the particle size distribution is very well controlled with these particles to be able to provide columns that have a very high reproducibility and robustness. So for this research work, we first took the anion exchange column and were able to separate full and empty viral species using this technology, which is resulting in a very nice baseline separation of both species using a very simple isocratic method. For the second part of the study, SEC column was used to determine the aggregation content of the viral vectors to analyze the monomers from the higher molecular weight species. Both fill state and aggregation are critical quality attributes to the adeno-associated viruses and need to be monitored rigorously.
Was there anything in your research that surprised you or stood out as a key insight?
So we did use for the analysis of the fill state. A salt gradient was used to determine between the full and the empty species. And for this purpose, it has actually turned out that it is very beneficial to include an isocratic hold step within that. So if you're going from the standard gradient to a gradient with isocratic hold, you can actually improve the separation between both species, which can be very beneficial for the analysis.
And how could your findings be useful for others in the field or even in real-world applications?
So by leveraging this monodisperse inert column technology, people have a set of columns available which they can use for the critical quality attribute analysis of adeno-associated viruses.